So someone recently pointed out to me that over the course of my career, I've given over 350 invited talks, and that isn't including internal ones like this one. But I've never before talked to a department of epidemiology, so I thought when I got this invitation, what should I talk about? And then I thought, oh, well, of course, I'll talk about the 150-year-old woman. Now, this is a topic of not casual, but very pointed interest to me, and that's because, to me, this is quite literally a $1 billion question, and so let me explain that. In the year 2000, I was invited to a symposium, a small symposium actually at UCLA of about 12 or 15 people on called The Future of Human Longevity. Small symposium on a big idea. And in those days, if I got invited to small symposia on big ideas, I would accept. And a few years previously, I'd gotten invited, for instance, to a symposium on the evolution of language, something that I knew nothing whatsoever about, which I told them, and they renewed the invitation. It was at a nice hotel in Geneva, so I decided I'm going to go anyway, and at least I'll learn something, even if they don't. And then right after that, this was my favorite of all invitations, I got invited to a symposium on international strategic initiatives in nuclear weapon deterrence, and something that I knew equally little about. And I found out, somewhat to my chagrin, that they took very seriously what I said about combat strategies and spiders and rattlesnakes, which made me worry about exactly where our nuclear deterrence policy was going. So when I got invited to this talk on the future of human longevity, I thought, well, at least this is something that I know about. I had already been studying aging for probably a dozen years at that point. And the nature of that was that we were speculating on how human longevity might change with the scientific interest in finding ways to slow the aging process. And we settled on a key length of life, which was 150 years, as something that we wanted to really focus on. Now, the reason we settled on 150 years is that we pretty much decided that traditional advances in medicine, better diagnostics, better treatments, were not going to get us to 150 years. They might get us to 130, maybe 135. That's for the longest live person. So we thought, there's no way that anyone will ever live to be 150 years old unless we actually figure out something about the science of aging and learn how to manipulate it. So at that, somebody asked the question, well, when will we have the first 150-year-old person? And when I was an undergraduate, when the professor would ask a class, I was always the one that would blurt out the answer, whether I knew it or not. So I blurted out, I think that person's already alive. And one of the people that was in the audience at that was a reporter from the New York Times, Gina Kolata, and she subsequently published in the New York Times what I had said. And here's what she published. She published a quote from me, and a friend of mine, a demographer named J. Olshansky, read that quote and responded in his typical direct manner. And so said, not only has Austad lost his mind, but he called me up and he said, I'll tell you what, you don't really believe that. I go, yes, I do, Jay, absolutely. And he goes, well, let's make a wager. And I said, okay. He had a book coming out. I had a book that had just come out. We figured this would be good publicity. So here is the wager. It was only, at that point, a $500 million wager. So we are academics, after all, so we didn't have $250 million to put in an account. So what we did is, early in 2001, we each put $150 into an investment account and decided that we would leave it there and let it sit there until the year 2150. And in the year 2150, if a single person, only a single person had to do this, if a single person had reached the age of 150 and it was cognitively intact enough to have a conversation with you, then I would get all the money in the best case scenario. More likely, my kids or their kids would get all the money. And if not, his kids would get the money. So for a long time, this was our $500 million bet. Now, the way we came up with $500 million is that the traditional rate of growth of the stock market, that $300 in 150 years would translate into $500 million. By the way, Jay has been managing that account. It's doing quite a bit better than the traditional rate of the stock market. So now, not long after I moved here to UAB, this came up time after time after time after time. Jay and I have done a sort of back and forth in front of all kinds of audiences. Never epidemiology departments, though. In 2016, a reporter goaded us into doubling the bet. So we doubled the bet. We each put in another $150. And so now it would be a nice even number of $1 billion. So what I'd like to do today in my talk is really explain why I'm going to win. So there is a possibility, of course, that I've already won. If you read the popular literature, here's an old article from Life magazine from the 1960s, where at this remote village in the Caucasus Mountains in Azerbaijan, there was a man, Sherali Muslimov, who claimed to be 161 years old. In fact, when he died a few years later, it was widely reported in the Soviet press, because this was such a good example of Soviet superiority, that he had died at the age of 165, and then a couple of years later he died again, I guess at the age of 167. Now, interestingly, not long after that, National Geographic gave a Harvard professor a lot of money to examine these places of exceptional longevity around the world. And so Alexander Leaf, who I knew when I was at Harvard, in fact, he served on the graduate committee of one of my students, visited 3 places. He visited this village in Azerbaijan. He visited a place in the Karakoram Mountains, which is kind of on the border between Afghanistan and Pakistan. And he visited a place in South America, Vilcabamba in Ecuador, the Southern Valley. And all of these things had people who claimed to be exceptionally old, not any older than Sherali here claimed to be, but 130, 140, 150. So I'm going to now let you in on a little secret. And that little secret is how you can live to be 150 years old or older. So here you are, and here is something that came out of Vilcabamba, the Vilcabamba Diet. You can also buy Vilcabamba water for health. But the key features are these. First of all, you need to eat and drink right. I feel good saying that in a school of public health. It helps if you live a lifetime of honest, hard, physical toil, and you keep on doing it, even in advanced age. You need to really live in a remote mountain valley of some sort. And here's the other thing that was odd. All of these reports of these oldest people were all men. So it helps to be a male if you're going to live to be 150 in one of these remote places. But the most important thing was that you needed to live in an area where birth records were unreliable, easily altered, or non-existent. In fact, what happened is that Leaf got the idea that he was being flummoxed when he went back. He visited Vilcabamba, and the oldest man in the village there told him he was 127, and he went back 4 years later, and this fellow had aged by 11 years in those 4 years. So he knew something was wrong. So, okay, that's bogus stuff. Let's talk about reality. How long can the longest lived people live? And here's a list of the 10 longest lived people for which we have reliable birth records. And you can see most of them are 116, 117, but there are a couple of outliers. There's Sarah Knaus, lived 119, and the longest lived of all, Jean Calment, lived to be 122 and a half, basically. Now, there's a great deal of, let's say, exaggeration, a great deal of hopeful exaggeration in these, even among these fairly well-documented people, and people are always being added to this list and then dropped off as questions come into account. And so fairly recently, Lucy Hanna, turns out, was only 115 when she died. But here's the thing that I want you to take away. Let's assume that the longest life is 122 and a half. That was Jean Calment. And we have more than 50 documents that validate her age. She really did live this long. The thing I want to note is that's the longest live person we know about. That's 22% shorter. So if we could increase that length of life by 22%, then someone would live to be 150 and I would win my bet. So let me chart out my path to victory for you. Here it is. So 1997, Jean Calment reaches the age of 122. By the way, Jean Calment played a role in my book. I got interviewed on the Today Show when my book came out. And at that time, that was just before Jean Calment died. And I said, she had met Van Gogh when she was a teenage girl in southern France. I said, if you hurry, you can shake the hand that shook the hand of Vincent van Gogh. And it was supposed to be a live interview because somebody famous died that day, got delayed for several weeks, and in the meantime, she died. So here they have this recorded interview where I'm talking a lot about if you hurry, you can shake the hand that shook the hand, and you could no longer shake the hand that shook the hand. So they had to edit it, and they edited it in a crazy way that made me sound like I was partially demented, which didn't help my book sales all that much. So, also in 1997, the highest life expectancy in any country in the world was 84 years, Japanese women, and the ratio, here's the key thing, the ratio, if the average, which is life expectancy, is 84, and the maximum is 122, that's a ratio of 1.45. What that means is that if you extrapolate to 2150 when my bet comes due, and 150 is what somebody has to reach, and 1.45 is the key ratio, then the life expectancy by 2150 would have to be almost 104 years. Now, some people think that that's crazy. J. Olshansky thinks that's crazy, for instance. But you know, we've had this remarkable increase in life expectancy over the last 170 years at least. Here's what it looks like for the United States. Since 1900, so in 120 years, life expectancy has increased by roughly 30 years. That's absolutely remarkable. If you calculate how much that is, it turns out that that's an increase of 6.5 hours a day for 120 years now. So calculate if you make it to the end of the day, your life expectancy is increased by 6.5 hours. Now, more recently, a group of demographers in Scandinavia published a paper where they said if you were born in the year 2000, extrapolating from current trends, then you could expect to live, notice Japan, 104 years. This means I'm going to win my bet if they're right. So as soon as I started thinking about how to spend the money, or more likely how my children or my grandchildren would spend the money, I started getting interviewed by people, and they would ask me questions about who's the winner? What's the winner going to look like? And so I said, well, the winner is going to be a woman. She's going to, because women basically live longer than men everywhere at all times. 90+% of people over 110 are women. As you saw, all the top 10 were women. Moreover, it's likely to be a Japanese woman, because Japanese women are the longest lived women on the planet. And not only that, 3 out of the top 10 women, oldest women, are Japanese women. And so I thought that's who it's going to be. It's going to be a Japanese woman. There's the 3, by the way, that made the top 10 oldest list. So I'm not really relying on this historical extrapolation because the low-hanging fruit for increasing longevity has been plucked. That was antibiotics, that was better hygiene, that was public health measures. Maybe recently a little bit of it is also medical interventions, but I'm confident because I'm very confident that we are going to find ways to actually slow the human aging rate. And let me give you some reasons why I'm so confident about that. First of all, it's not because of nonsense like this. It's not Dr. Oz and Gwyneth Paltrow and this kind of nonsense. Why I'm so confident really has to do more with the fact that we're understanding more and more and more about the biology of aging. Since I got into the field, which was in the late 1980s, I'd say the rate of progress in understanding aging has accelerated to an incredible extent, so that when I teach the biology of aging, which I'm doing this semester, for instance, I tell them at the start of the semester, before the semester's over, there's going to be some important breakthrough that we need to talk about, because that's how fast things are happening. So we understand a great deal now about the biology of aging. We understand a lot about the cell biology, about places in the cell and biochemical pathways and networks that need to be tweaked. For instance, we know that the mTOR pathway is important. We know that insulin IGF signaling is. We know AMPK is involved. We know the mitochondrial function and mitochondrial communication between the mitochondria and the nucleus is important. And in the top one that's circled there, some senescent cells. This is the hottest thing in biology right now, in the biology of aging, is senescent cells and understanding what we're going to do about them. So because of the Gwyneth Paltrows and the Dr. Oz's and the other charlatans out there that had been saying that people could live longer, stay healthier by taking all this stuff, the National Institute on Aging decided to put together a program to test products. And they put together a program called the Interventions Testing Program. And originally, that program was designed to try to debunk all these bogus claims that was out there. But while they were putting it together, and I was at the original symposium, the original small meeting where we designed it, while they were doing that, we were learning more and more and more about aging, so that by the time they put the interventions testing program together, it actually turned out to be incredibly successful. Let me just tell you a little bit about the design, because that's important. First of all, they'll test any compound that you can suggest to them has a chance of working. And they will test it for free, and they will put your name on any publications that come out of it. And in fact, some people from UAB suggested a drug called acarbose that actually turned out to work basically in one sex, and they've been now involved in a number of papers. But here's the other thing: if it doesn't work, they're going to publish that too. And it's done in 3 different places, identically, so you have instant verification by 3 places. And the thing that was surprising is, lo and behold, did they not only not start debunking drugs, they started finding drugs that actually help. Now the other thing is, this is in mice. It's an unusual kind of mouse. It's an outbred mouse, but it's in mice. So before the ITP was very old at all, in their second group of compounds that they tested, they found one that hit the bullseye. And that drug was a drug called rapamycin. And it was called rapamycin because it was isolated from a soil bacterium on Easter Island. Easter Island is Rapa Nui in the local language, therefore rapamycin. Now, it wasn't just that rapamycin made mice live longer. I mean, we've been able to make mice live longer by reducing their diet for 75 years at the time this happened. Let me give you 7 effects of rapamycin, and all of these, except for one, have been done in multiple laboratories, so it's not something that only one laboratory can ever reproduce. First of all, rapamycin at a wide range of doses, a range of doses of at least tenfold, extends life. It does it very robustly. It does it in every kind of mouse in which it's been tried. The other thing it does is it completely prevents or else slows the development and delays the onset of other types of cancer. You probably know that laboratory mice, by far the majority of mice die of cancer. So this was important, but that wasn't the only thing. It delayed the development of mouse versions, genetically modified of atherosclerosis and normal late life dysfunction. It turned out that rapamycin rejuvenated stem cells of a whole wide variety of stem cells. It also turned out to enhance and broaden vaccine protection. And this was particularly intriguing and unexpected, I would say, because rapamycin has a clinical reputation as an immunosuppressant. It's used as part of a combination, immunosuppressive combination for kidney transplants. But what it really is an immunomodulator, and in some ways, in some instances, it's actually an immune system stimulant. And the interesting thing about the vaccine protection is that it not only worked in mice, but there's been a trial in people, and it worked exactly the same way in people as it did in mice. Not just the mouse work was to the flu, but they've subsequently looked at other infectious respiratory diseases. And again, notice that all of these things, I usually don't put scientific citations on slides like this, but I just did it to emphasize that in virtually all of these cases, multiple laboratories using multiple approaches have all found similar things. A number of labs using different mouse models of Alzheimer's disease have found that it delays the onset and slows the progression of mouse versions of Alzheimer's disease. And then something that's only come out recently, when a friend of mine hired a dental student to actually work in his lab, it turns out that rapamycin also prevents or slows the progression of periodontal disease in mice. So if you look at all these things, what you're looking at is something that has broad effects on age-related diseases, something that I think most of us would say, yes, there's evidence that actually is delaying aging, at least in mice. We don't know about people yet. Now, to emphasize how much we know about aging, here's a list of some of the other drugs that the Interventions Testing Program has tested. And I've only pointed out the ones there where the effect is big enough to make me win my bet if it works in people. And most of these, we've only looked at one or a couple of doses, so it may be that when the doses were optimized, the effect would be even better. So, a couple of other things about rapamycin. The other thing is that rapamycin had major effects even when it was started late in life. When I got into the field, the one thing that you could rely on was if you took a mouse or a laboratory mouse or rat, cut back the diet, they would live longer. But it didn't work after a certain age. It only worked up to about midlife, and then if you started it after that, the effect went away. Turned out with rapamycin, the effects are substantial. In fact, they're almost as big later in life than they are earlier in life. And in fact, the original paper, what they usually say about that paper is that it increased longevity by about 13% in those animals. Turns out that's not really true. That was because they didn't analyze the data in a sensible way. They actually didn't start those mice on rapamycin until they were 20 months old. But then what they looked at was the effect on the entire lifespan. If you looked at the effect on lifespan from the age at which they started getting the drug, the effect was 38% in females and 28% in males. So that's why I'm not too bothered by the fact that we don't have anything today when the people who were born in 2000 are now 20 years old. They can still start in the next few decades, and I'll still feel confident. Well, all of that stuff is about mice, right? What about people? So a couple of friends and I got a grant a few years ago. It's called the Geroscience Network. And the idea of the Geroscience Network was to bring together basic biologists in aging, physicians, pharmaceutical experts, and try to come up with a experimental plan to test these drugs. And so these are the three of us, Jim Kirkland from the Mayo Clinic and Nir Barzilai from Albert Einstein. And what we did is we rented this castle in the middle of nowhere in Spain, this 15th century castle. And we got about 20 of the smartest people we knew, and we trapped everybody in that castle for about 5 days to design the study. The key study. Now, I wanted this study to be about rapamycin. I thought that would make a lot of sense given what I've said, but I was eventually talked out of that into a different drug. So here's the drug that we decided that should be the first human trial. It's for the historical records. If you had type 2 diabetes, your life was shortened by order of 6 to 10 years if you didn't have good glucose control. There was a published study in 2014 that compared type 2 diabetics on this drug to non-diabetics that were matched for age, socioeconomic status, sex, all the standard things. Turned out that drug worked best if people started taking it over 70. And numerous, here's why, epidemiological studies suggested that drug had some effectiveness at preventing or delaying the progression of cancer, cardiovascular disease, and dementia. And so this is the drug, metformin, the most commonly prescribed drug for type 2 diabetes in the world. And so at that, while we were isolated in that castle, we really came up with a study design, one that I thought that we all felt we could, within 5 or 6 years, we could determine whether that drug actually slowed multiple diseases and preserved function that is slowed the process of aging. So in the time since then, we have struggled to find the financing for that, because one of the nice things about metformin is it's dirt cheap. It's off patent, which means that pharmaceutical companies aren't going to fund it, but we finally come up with about the $65 million needed to start this trial. It's called the TAME trial, targeting aging with metformin. And we actually presented this design to the FDA. And we said to the FDA, if we design a study like this and we show these effects, would we actually get approval, FDA approval? We couldn't say for treating aging, because they don't believe in aging, it's not a disease, they only believe in diseases, but for treating multiple features of multiple diseases. And they said they would. So that trial is about to start. And it's going to be very interesting. None of us at the time really thought metformin was the be-all and end-all drug. We just thought this is something that the safety profile is very well known. It's dirt cheap, so the FDA is not going to suggest that we're suggesting it only so that we can make a pile of money. But there's a whole bunch of other emerging possibilities. That's just one. There's probably going to be better drugs. We're going to learn more about dosage, how often to give them and all. Let me just talk about several other possibilities, though. Starting in the early 2000s, people took a very old technique, which was sewing two animals together. It's called Parabiosis, and allowing their blood streams to merge. And what this new study did, Tom Rando at lab at Stanford started this. Is everybody getting a weather warning? So maybe my talk will get, it's a flash flood warning. We're on the 4th floor, who cares? Right? So what Rando did with this old technique really was say, well, what would happen if we sewed an old mouse to a young mouse and let the old blood and the young blood commingle? And what they discovered, his original work was on muscle, and it had an enormous rejuvenating property on damaged muscle. It's now been done a whole bunch of times, and it's now known to rejuvenate multiple organisms, not just muscle, but brain, liver, heart, and it improves the function in multiple organs. Now, one thing that they don't point out is it makes the old mice seem younger, but it makes the young mice seem older. So I always tell my undergraduates, there's a business opportunity here for you guys. How much do you sell your plasma for? Because it turns out that I sold plasma when I was in graduate school, and I think I got about $50 a pop. You can now get about $8,000 for blood if you're 25 or under. It turns out that if you do this in germ-free, or in mice that have had their immune system suppressed, plasma from young humans also rejuvenates the organs of old mice. And so there's a whole bunch of current research aimed at understanding what it is in the blood of the young mice that's rejuvenating, or possibly what there is in the blood of the old mice that's actually toxic. And there's been a few false starts, let's say, in that business. A few years ago, a group from Harvard thought that they'd found the key ingredient in this blood, but nobody's been able to repeat that. So right now we're back to there's something, but we don't know what it is. Again, one of the reasons to believe this, it's been found in a number of independent labs. It hasn't just been found in one lab. The most recent thing to occur was zombie cells. Senescent cells are zombie cells. And Alex Carrel, who's shown here, thought for years that if you took cells out of the body and grew them in a dish, that they could just go on forever and ever and ever. And in the early 1960s, this fellow here, looking as grumpy then as he still looks, Len Hayflick, discovered that that's not true. That normal human cell, that's true of cancer cells, but normal human cells, if you take them in a dish, they will divide a certain number of times and then they will stop. And he called that cell senescence, and he thought that it was a model for aging at the whole organism. And so for the longest time, there was this contention in the field. And I remember going to symposium after symposium where people were arguing about, is this really teaching us something important about aging, or is this only an artifact of cell culture in a dish? Well, it turns out that there's something real there. There's something very real about these cells, and they do a lot more than simply stop dividing. In fact, these kinds of cells have a good side and a bad side. They do stop dividing, and that's thought to be a cancer prevention mechanism, basically a tumor suppression mechanism, because if a cell can only divide a certain number of times and then it stops, it's protected from ever forming a massive metastatic tumor. And I got my friend Judy Campisi up here because she's really discovered a lot. Most of what we know about the phenomenology of senescent cells she has discovered. It also, cell senescence turns out, is incredibly important during development. The way that your brain cells are pruned when you're developing, the way that the webs in your extremities are pruned, that's all has to do with cell senescence. But it also has a bad side. And the bad side is not just that the cells can no longer help you repair a damaged organ. It turns out that these cells ooze this toxic material. She called it the SASP, which is the senescence-associated secretory phenotype. And it was that SASP that was thought to be causing a lot of the problems in older individuals, because even in older individuals, it's a tiny, tiny percent of all the cells that ever become senescent. And so what's happened recently is that we have discovered ways to get rid of some of these senescent cells. In a 2016 paper, it was really shown you had to genetically modify the mice and then give them a drug. But the fact is, when you did that and you eliminated a number of the senescent cells, you all saw all kinds of health benefits in older mice. A couple of years later, the first drugs were identified that specifically targeted senescent cells and not normal cells. And by the way, this is a mouse model in which the senescent cells light up, and so this is mice that are treated with vehicle, and these that are given drugs that this is dasatinib, this is quercetin, that actually eliminate senescent cells, and you can see by comparing these how much the senescent cells are eliminated in these mice. And more recently, there's been a paper that showed in very old mice, survival was increased by 36%. Now I'm thinking 36%. I still only needed 22% to win my bet. Interestingly, what we found most recently is that they've already started some small human trials. And what we've found so far is not an effect on health, but what we found is a lot of evidence that the same drugs that they're giving to mice, when you give to people, do in fact eliminate senescent cells. The health effects of that we're not so certain of. There's 2 studies, one in IPF and one in diabetic nephropathy, that show some promise, but they're really very small, very preliminary, but stay tuned. So those are all the reasons that I think I'm going to win my bet. So imagine my surprise. Now I've made this bet. I'm feeling very good. My kids are in the best case scenario. I'm going to be fabulously rich at some point. And then in 2002, I got a phone call from a fellow who told me that he was the chair of the President's Council on Bioethics, and he wanted to talk to me about the ethics of what we were doing. And it never occurred to me, because I'm a scientist, it never occurred to me that anybody would have any thought except this is the best thing since sliced bread. So then I started to worry, well, what are they going to do? Because this council, for those of you who don't remember it, was put together by President George Bush to kill stem cell research. And once they finished doing that, they decided to move on. And the next thing on in their sights was aging research. So I started thinking, well, is this such a good idea? And what are the issues involved? And what really disturbed me is after I testified, I was the only biologist called to testify to the President's Council. After I did this, I started getting invited to bioethics meetings, even though I didn't know anything at all about bioethics. So another one of these feels like nuclear disarmament that you can be an expert in by giving one talk in one venue. Kind of frightening. So let's consider that now. Let me just finish up by considering several aspects of it. This, by the way, is what we've done since the year 2000. We have dropped the age-adjusted rate of death of all of these things, stroke and heart disease, by over 30% just since the year 2000. All of that is very, very, very good. But that's not the only thing that has changed since the year 2000, because there are side effects. There are things that happen when you improve, when you keep people from dying that you may not wish to do it. So nursing home residents has increased dramatically. Diagnosis of Alzheimer's disease has increased dramatically. Various indicators of what I think of as late life misery are all around us. They're around us so much we don't even think about it anymore. How many people do we know that have had to have hip replacement or knee replacement because they're in insupportable pain? Also, we've had a 300,000 year go as Homo sapiens. That's how old our species is. We've never lived remotely as long as we do now. And what that means is that as we continue to age, as the population continues to age, we're in for a lot of what you might call bad news, and that's not even to mention the massive healthcare costs that are going to assume that. So some people consider all of this stuff very, very bad. So this is, I think, where the President's Council on Bioethics is going. By the way, I thought that I had changed all their minds on this when I gave my testimony, but then they came out with a book a little bit after that, and I found out I was wrong. So why is there this ambivalence about it? I bet people that work on heart disease or cancer or Alzheimer's disease, nobody ever says, you really ought to not do that. You know, there could be some ethical problems. What's going to happen if we cure cancer tomorrow and everybody lives longer, and then they're going to get osteoporosis and sarcopenia and all? But in the aging business, we have to put up with this a lot. And I think the reason is pretty obvious. If you imagine our current lifespan as having this period of good health and then a gradually increasing period of ill health, I think the way that most people think about what we're going to do if we make people live longer is this. they're going to live longer, but they're just going to live longer in this state of compromised health and debility, gradually increasing debility. And I think nobody wants that. So that doesn't seem to be the case. In none of the ways that we know to make our experimental animals live longer is this actually happening. Are we really just keeping them alive longer? So one of the arguments that I run into is that we are somehow violating the natural lifespan when we do this. That this is somehow compromise our humanity. Well, you know what the natural human lifespan looks like? It looks like this. It looks like about 20 years, and that's because half of the kids are dead by 10 years old, and the adults are sick for most of their very short life. And I used to work in New Guinea, and I've seen this impossible. In fact, these are some of my friends in New Guinea from when I worked there. So I think that, first of all, the natural human lifespan is long gone. Good riddance. I think everybody would agree. Good riddance. We don't want to go back to having a life that's nasty, brutish, and short, as Thomas Hobbes would say. But there's also an issue of fairness. Is it going to be fair if we invent these ways to slow aging, prevent diseases, if it's not available to everybody? Because quite likely it won't be available to everybody. But you know what? We already have a fairness issue. Look at this. This is life expectancy at birth as it's changed over an 18-year period, and people with less, this is in America, people with less than a high school education, and as you can see, it's actually gone down by 5 years. That's massive. That's the difference between the United States and Bangladesh, roughly. On the other hand, things are very good for those of us with a college education. So fairness is already an issue. It's not just that. Look at the diseases of people from low, medium, and high education. We're basically already living as basically almost separate species. There's the healthy, highly educated, wealthy people and the other people. And this is even extended. Is anybody paying attention? We have 5 candidates for president that are going to be over 70 years old when they're inaugurated. But because they come from this upper socioeconomic strata, their 4-year survival probabilities are really quite remarkable. In fact, if you calculate their life expectancy, all of these people have a life expectancy that would take them well out beyond 2 terms as a president. So we already have fairness issues. Those are issues that if we're going to deal with them, we're going to need to deal with them whether we have advances in preserving health or not. What about resource issues? What about the environment? What about the cost of taking care of these older people? Thomas Malthus, as you may recall, is a person in the 17th century that said that human population growth is going to outrun the food supply, and we're going to have all this misery as a consequence. Didn't really happen then. Maybe happening now, we don't know. But I think what we need to think about that is that these issues are going to need to be addressed, whether we slow aging or not. And I think I'd rather address them with a bunch of older people who are in good health rather than bad health. Let me just show you this. This is a direct quote from one of the people that thinks we ought to worry about this a lot. The burden of supporting old people is too heavy. Politicians are disturbed by the enormous cost of the state support of the aged, and neglecting the fact that politicians themselves are not the youngest people on the planet. Another statement from the same person: the burden of maintaining the aged will become even heavier. People continue to live longer. The resources of the young will suffer. So tenure track faculty positions will become rarer and rarer as people hang on. This was actually said by Ilya Mechnikov in 1907. In 1907, the life expectancy in France, where he lived at the time, was 50 years of age. And somehow we've muddled through. For another 113 years, I think most of us would agree that we'd rather be alive now than in 1907. And so finally, There are a number of other scenarios, and we're really not sure. There's the current scenario. There's a scenario most of us wish for, which is that we would extend the period of healthy life without lengthening the period of debility towards the end. Or even if we don't do that, if we could just compress the morbidity, we'd be happy with that. These are all good things. In the best case, we extend healthy life and at the same time compress morbidity. And here's one I call Socrates' nightmare, and this is the one that we're currently facing. And that's that we will basically increase the period of healthy life, but also increase the period of ill health at the end. That's where we are right now. Life expectancy is still increasing, but it's not increasing as fast, but health expectancy is not increasing as fast as life expectancy. And I think that we're going to have to make some decisions there. These are issues for philosophers, not for biologists. I just present it as possibility. So here's one thing I'd like to point out in closing. We've already made a decision that delaying, preventing late life diseases is worthwhile because we don't encourage smoking. If we wanted to, if we were worried about preserving health, we would probably say, well, we really want to see people smoke as much as possible. We don't, most of us don't, renounce vaccines and antibiotics. We don't destroy insulin supplies. We don't reward reckless dieting. And if an old folks' home catches on fire, we don't say, I'm sorry, they're going to be too costly. They're not productive anymore. Let's just let them burn to death. We rescue them, and we make heroes out of the people that rescue them. Furthermore, we try to prevent and cure cancer. We try to prevent and cure heart disease, Alzheimer's disease, diabetes, COPD. You have it. And all we're talking about with aging is trying to do all of these things as a group at the same time. Who could be against that? Well, ask Leon Cass, who was the chair of the President's Council on Bioethics. So let me just summarize at the end here. So the goal for the effort to slow human aging is not just so that I will win my bet. That's just a fortunate side effect. The goal is to preserve health and not necessarily to prevent death. I would be almost as happy if we could just compress morbidity. That goal is absolutely consistent with everything that we do in biomedical research. We're just trying to do it in a more generalized and robust way. One thing I haven't talked about is that addressing these things one at a time could turn out to be a nightmare. If we cured cancer and heart disease, it would extend human life expectancy by about 15 years, but we wouldn't be protected against osteoporosis, we wouldn't be protected against Alzheimer's disease, sarcopenia, frailty, all kinds of other things. So there is a rationale for addressing these things as a group. And finally, treating aging itself, the underlying process, is a more effective approach because it preserves health and treats multiple diseases at the same time. Ultimately, what we would like is for everybody to be able to look as good and feel as good as this 100-year-old and his 70-year-old son. This is kind of cheating because this is genetics, but we think that we could do the same thing with some medications that will be around soon. Thanks for your attention.